Hi all. Now we will see the classification of viruses based upon the presence of a lipid envelope. I have already told you that some of the viruses only carry the lipid envelope, and these viruses which carries this envelope is known as enveloped viruses. Envelope is actually lipoprotein molecule which is having a lipid covering, and which on the surface of this lipid envelope we can see protein spikes. Protein spikes is otherwise known as peplomers. And whereas the viruses without this envelope is known as non-enveloped viruses or naked viruses. Now we will see another system of classification based on the host cell preferences. I told you before that the viruses can attack different type of a host like plants, animals, humans, bacteria, etc. Even within a host cell, they have got particular host cell preferences. So based on their host cell preferences, we can classify viruses into different types. Among this is a dermotrophic. Dermotrophic viruses are those viruses which infect skin cells. For example, you can see certain type of herpes viruses and also certain papilloma viruses which will infect the skin cells. Whereas another second type is neurotrophic viruses. Neurotrophic means the viruses which infect the nerve cell. Example is rabies virus. Then another third one is the viscerotrophic viruses. Viscerotrophic means which infect the organ of digestive system. Digestive system that infects the digestive system. Example is rotavirus as an example, and also poliovirus. And the fourth one is pneumotrophic, which infect respiratory system. Examples include the SARS virus, the COVID, the very notorious COVID virus, etc., which infects the respiratory cells. So this is another method of classification of viruses. Now all of you know that based upon the genetic material, we can broadly classify viruses into DNA viruses and RNA viruses. DNA viruses are those viruses which consist of DNA genome, and they complete their activity by transcription, and most of them attack on organisms of similar genome, similar type of genome, DNA genome. And RNA viruses, which are the viruses that carry the RNA genome, they complete their activities by using the process known as reverse transcription. By using the reverse transcriptase enzyme, they convert the RNA into DNA and they perform the activities. Now we will discuss about the most important system of classification of viruses, that is based on the type of genetic material and also how the viruses uses this genetic material to synthesize their mRNA and viral proteins, and for the assembly of this viral proteins into mature infectious viruses. So it is actually introduced by scientist David Baltimore. And here I repeat, the classification is based on the genetic material, that means whether they carry DNA or RNA. In the DNA itself, whether it carries single-stranded DNA or double-stranded DNA. In the case of RNA also single-stranded or double-stranded, and how they use this genetic material to synthesize the mRNA. So when they attack a host cell, they need for the multiplication they need to produce viral proteins, and these viral protein assemble together to form a mature virion. For that they have to synthesize the viral mRNA, and also the production of viral mRNA, and also from the viral mRNA it need to produce viral protein, and this viral protein need to be assembled to produce the new virus cell. And how these activities are conducted, and then this classification system is done. And based on this Baltimore system of classification, viruses can be classified into 7 groups. So we'll discuss about these 7 groups. The first group is double-stranded DNA viruses. The double-stranded DNA viruses are the viruses which carries the double-stranded DNA, and here the transcription and translation is similar to normal cells. Here from this double-stranded DNA, mRNA is synthesized, and this mRNA which produces the viral protein. This is a normal pattern of cell transcription and translation. Double-stranded DNA that act as a template for the production of mRNA, and this mRNA which produces protein. And examples of this class include the papovavirus group. The papovavirus group includes certain type of viruses known as polyoma virus, SV40, that is simian 40 virus. We will see about certain type of viruses later. Now we just know the names of this virus. Pox viruses are also included under this group. Example, vaccinia virus. And another group is adenoviruses. Example is human adenoviruses example. And then herpes viruses are also included under this category. Herpes viruses examples are herpes simplex virus type 1 and type 2 herpes simplex virus. And also another virus is known as Epstein-Barr virus. Epstein-Barr virus is actually the virus which causes a particular disease in human beings, that is infectious mononucleosis. So this is the first group, that is double-stranded DNA viruses. Now the second class is actually the viruses which carries single-stranded DNA. They carry single-stranded DNA. Here, in such viruses, this DNA, that's the single-stranded DNA, and another intermediate DNA is synthesized before the synthesis of mRNA transcript. That is, it produces a single-stranded DNA which is converted into mRNA. And actually the mRNA has the same polarity as the DNA, but actually it is complementary to the intermediate DNA. So once again I will explain, that is single-stranded DNA, this is a plus single-stranded RNA, this plus stranded single-stranded DNA. This plus stranded single-stranded DNA is converted into an intermediate DNA, and which produces the mRNA. And this mRNA has got the same polarity as that of the DNA because there occurs an intermediate. An example is parvoviruses. Now, the third group is the plus-stranded, single-stranded RNA viruses. The virus carries the RNA genome which is that's a single-stranded. Moreover, we have a designation here. This is a plus-stranded. But I will explain what is plus-stranded. Here plus-stranded means here this RNA has got the same polarity as that of the mRNA. That means that this single-stranded RNA itself acts as the mRNA. So there is no need to synthesize a different type of mRNA. Here this single-stranded RNA that itself acts as a mRNA. So it has got the same polarity as that of the mRNA. That is why we use this designation, that is plus-stranded. These viruses of this group can be grouped into two subclasses. First subclass is subclass 3A. Here the individual mRNA encodes a polyprotein or polypeptide which is broken later into form the viral protein. That means this single-stranded mRNA that produces a single polypeptide which is broken down to form the viral proteins. Example is picornaviruses. Picornaviruses that includes a particular important virus known as the poliovirus. Whereas the subclass 3b, that is actually the third subclass category B. Here this plus stranded RNA, that is two types of mRNA molecules are transcribed, from which two types of mRNA molecules are transcribed. One is as that of the same length that of the virion RNA, and other is a fragment of virion RNA that produces two fractions or two splits of mRNA molecule. Examples include Toga viruses, Toga virus group. There's the alpha viruses. Examples are Sync virus, Semilki virus, and Havi viruses, etc. And also the group B, that is the Havi virus group, that's dengue fever, yellow fever, St. Louis encephalitis virus. That certain arboviruses are coming under this group. Now, the fourth type. The fourth type is minor-stranded single-stranded RNA viruses. Here, the viruses has got an RNA genome which is single-stranded, but the sense is minus-stranded. Here, this minus-stranded means it is complementary to the mRNA. That means this itself cannot act as an mRNA. This acts as an intermediate, and that produces a mRNA which is complementary to this RNA. This group also has two subclasses, 4A and 4B. Here in 4A, the single-stranded RNA genome encodes a series of monocistronic mRNA. That is, this RNA genome that produces that produces as monocistronic, having a single cistronic mRNA. Examples include rhabdoviruses, mumps, measles, Sendai virus, etc. Whereas in the subclass 4b, each segment molecule of the genome acts as a template for the synthesis of an mRNA which are monocistronic or encodes a polypeptide. That is, here the genome is segmented, and this segmented genome acts as a template for the synthesis of mRNA, which is also monocistronic in nature, and that codes for a polypeptide. Here the difference is that in subclass 4a, this single-stranded RNA genome is continuous and it codes for a monocistronic mRNA. Whereas in the subclass 4b, it is segmented RNA and which produces monocistronic mRNA from each template. Examples of this group is orthomyxoviruses. Example, human influenza virus. You might have heard about human influenza viruses which can undergo genetic rearrangements and which undergoes genetic mutations, etc. And in the cases, most of these viruses usually the different variants will appear due to this mutation or genetic rearrangement because the genome is segmented. We will study about influenza virus later in the second module in detail about influenza virus. Then you will study more about its segmented genome and genetic rearrangement. So this is the minor-stranded RNA viruses. Okay, this is single-stranded RNA which is minor-stranded, which is divided into two subclasses. One is unsegmented, other one is segmented. Okay.